Hi everyone, my name is Diane Ibarra. I am a second-year graduate student in the Experimental Psychology PhD program, and my mentor is Dr. Wambo Peng, and today I'm going to be sharing with you my poster titled ECT-Induced Memory Disruption: Insights from Local Field Potential Recordings in Rats. So, electroconvulsive therapy, or ECT, is a procedure that is used to treat severe mental health conditions when prior treatments have failed. While ECT remains as a last resort intervention, this treatment does yield the most effective outcomes. However, studies have found that the use of ECT has led to memory impairments in some patients. In this exploratory study, we are using ECT to test if a painful memory can be eliminated through the use of continuous local field potential, or LFP recordings, and through the formalin inflammatory pain model. For the methodology, a total of 9 adult male Sprague-Dawley rats were used in the study. 6 were in a formalin-only condition, and 3 were in a post-formalin ECT condition. With the animals under anesthesia, 4 electrodes were intracranially implanted into the target brain areas, being the right anterior cingulate cortex, or ACC, the right central amygdala, the right CA3 hippocampal region, and the left CA1 hippocampal region. A wireless recording device was connected to the electrodes, and a USB dongle was used to transmit the signals onto a computer. LFP was recorded under 1.5% anesthesia isoflurane for both conditions. In the formalin-only condition, or Group A, after a 10-minute baseline, 50 microliters of 3% formalin was injected into the left hind paw of the anesthetized animal, and LFP was recorded for a total of 65 minutes. In the post-formalin ECT condition, or Group B, after a 10-minute baseline, 50 microliters of 3% formalin was injected into the left hind paw of the anesthetized animal, and LFP was recorded for 20 minutes. After the 20 minutes, 3 ECT stimulations at 50 milliamp, given 12 to 15 seconds apart, were delivered. LFP was recorded for an additional 35 minutes. For the results, Figure 1 displays 3 power spectrograms for 3 representative animals and 2 power spectrum analysis summary graphs. Figure 1A shows a power spectrogram and a raw wave trace of the CA3 region of a representative animal from the formalin-only condition. Figure 1B and Figure 1C also show a power spectrogram and a raw wave trace of the CA3 region from 2 representative animals from the post-formalin ECT condition, showing facilitation and inhibition after the administration of ECT. The red arrow represents when a formalin injection was given, and the blue arrow represents when ECT was administered. Figure 1D shows summary graphs for the post-formalin and formalin-only groups. The columns represent the 4 brain regions, being ACC, right central amygdala, CA3, and CA1, and the rows represent the 5 frequency bands, being delta, theta, alpha, beta, and gamma. In the formalin-only condition, there is an obvious formalin response when the formalin injection is given at 10 minutes, which is represented here by the red arrow. In the post-formalin ECT condition, there is also an obvious formalin response when the formalin injection is given at 10 minutes. However, there seems to be a mixed response after ECT is given at the 30-minute mark, which is supported by these 2 power spectrogram analyses here, where there is facilitation and inhibition in the same group after being given ECT. Figure 2 displays 5-minute data with Group A being in orange and Group B in blue. Due to the small sample size, statistical significance was only observed in the beta frequency band of the CA1 region at 50 minutes. Once again, the post-formalin ECT group shows both inhibitory and facilitatory effects after ECT. So in conclusion, the LFP results indicate that ECT elicits a mixed response when administered following acute inflammatory stimulation. Further research is needed in a larger sample size, and also needed in unanesthetized animals to come to a better conclusion. Thank you.